Okay, so here we are moving along. We've mastered the amino acids, hopefully, and we've started to learn more about proteins. We've described the primary and secondary levels of protein structure, and you're ready to move forward with more. All right, so specifically in this lecture, we're going to focus on tertiary and quaternary levels of protein structure. So just when you thought it couldn't get any more complicated, we're ready to add some more twists and turns, quite literally sometimes, to protein structure. Okay, so if you remember, the primary structure was the sequence of amino acids, and then the secondary structure is what starts to happen when these amino acid residues start to interact with each other, while the tertiary structure of a protein gives a specific three-dimensional shape to the polypeptide chain, and it involves attractions and repulsions of the R groups of the amino acids of that peptide chain, stabilized, of course, by things like hydrophobic and hydrophilic interactions. So we mentioned that some side chains are hydrophobic and others are hydrophilic, and that's going to allow them to interact positively or negatively. We're going to have salt bridges possible. We'll talk about that throughout this lecture. Hydrogen bonds, and then finally cysteine residues. We've already been warned will make these disulfide bonds, and those are critical to the tertiary structures of proteins. It's also important to understand that you're likely to find multiple types of these interactions. Not necessarily all of them, not every protein is going to have disulfide bonds perhaps, but you're likely to see some mix of hydrophobic, hydrophilic interactions, salt bridges, disulfide bonds, in addition to, of course, the secondary structure features we've already mentioned, like the beta pleated sheet, the alpha helix, and the triple helix. Okay, so since we've really focused on those R groups in determining the tertiary structures, right, the primary was just the sequence of bonding in the amino acids. The secondary was largely due to those amino and acid carboxylate groups interacting along the chains. Now with tertiary structures, we're really focused on the R group interactions. And so as a reminder, we've seen this before, right? At least most of it, that nonpolar hydrophobic interaction. We could have the polar and water hydrophilic interactions. There's the polar basic and the polar acidic. Those are what form the salt bridges that we mentioned as something new and different here in tertiary structure. Also polar neutral and polar neutral can form hydrogen bonds between the OH of the hydroxyl groups or the NH or NH2 of the amide functional groups. And then, of course, those cysteine residues are suspect for disulfide bonds. Not every cysteine is going to make a disulfide bond, but every cysteine you encounter, you should be thinking about the possibility of disulfide bonds. And of course, with tertiary structure, those cysteine residues can occur hundreds of amino acids later because the folding can allow them to be close enough for a disulfide bond where you might not think about that from the primary structure. We started the whole chapter with a discussion of some different protein types and protein functions, and one of the major types would be globular proteins, and globular proteins tend to have compact spherical type shapes. They're often used to carry out synthesis, transport, and metabolism in cells, and they would include proteins such as myoglobin, which store and transport oxygen in muscle. So here we see a picture of the folded tertiary structure of myoglobin. You've got the heme group, you've got the carboxy terminus and the nitrogen amino terminus pointed out for you. And you can see that it's a highly folded and roughly spherical packing for this myoglobin globular protein. If we look, on the other hand, at fibrous proteins tend to consist of long fiber-like shapes, so very different from the globular proteins. And you can have things such as alpha keratins, which make up hair, wool, skin, and nails. And there we see our sheep there with alpha keratin, which has the alpha helix as a prominent type of structure. We can also have feathers containing beta carotenes with large amounts of beta pleated sheet structures. So those secondary structures are important and do influence these tertiary structures, but it's really the interplay of secondary structure and side chain to get that overall tertiary structure type. Okay, so before we leave tertiary structure, let's have a learning check, make sure we're getting what we need to be getting from this particular topic. So here you're asked to select the type of tertiary interaction of the following. So A, we're looking at leucine and valine. B, we're looking at two cysteine residues. That should be setting off alarms. C, we have aspartic acid and lysine residues. And then finally D, we have serine and threonine residues. So for A, B, C, D, if you could match them up with 1, 2, 3, 4, just pause the video here if you need a moment to do so. Start us back up when you're ready to check your work. Good luck. Okay, so there we have it. Leucine and valine are two examples of non-polar neutral side chains, so therefore hydrophobic interaction would be the most likely type. B, two cysteines, hopefully you've already caught on that those are the likely disulfide bridge formers, or disulfide bond formers, as we use both terms. So that would be 1. C, aspartic acid and lysine. Those are our side chains that are ionizable. We have an acid and a base, so they can form C, hydrogen bonds, or ultimately those can form salt bridges, so they have ionic interactions. And then finally, with D, serine and threonine, those are examples of polar side chains that will not ionize. So we're not looking at ionic salt bridges here. We're looking at the hydrogen bonds. 3 here would be our major type of interaction expected from those residues. So hopefully that went well. If it didn't, hopefully our explanation has helped. But if you're still struggling, then certainly, as always, please reach out for some help. Okay, and here we go, quaternary structure. So this is the end, believe it or not. I know we seem to just add additional layers. Well, this is the last one we're going to toss on here. And quaternary structure is the combination of 2 or more polypeptide chains. So it's stabilized by those same interactions found in the tertiary structures, but it's different subunits coming together. So if we're looking at hemoglobin in particular, for instance, it consists of 2 alpha chains and 2 beta chains with heme groups in each subunit that pick up oxygen for transport in the blood to the tissues. So there we see the sort of pink magenta colors and the orange colors. Those distinguish our 2 different types of subunits. So we have the 2 alpha chains and the 2 beta chains, and they all come together in this quaternary structure to form the functional protein. Okay, so now that we've learned them all, let's see if we can make sense of this summary table here. So the primary structure, again, is just the peptide bonds joining the amino acids in their specific sequence in a polypeptide. Insulin, as we remember, was the first protein to have its primary structure determined, and that just told us the order in which they bonded. It didn't tell us necessarily about which residues were critical for the function of that protein. The secondary structure gives us information about things like the alpha helix, beta pleated sheet, or triple helix that can form by hydrogen bonding between the atoms in the peptide bond along the chain. So really we're looking at those peptide bonds. We're not looking so much at the side chain at this point. Tertiary structure, that's the polypeptide folding into that compact 3-dimensional shape. And this is where we're stabilizing with interactions involving those R groups to form something that's biologically active. And then finally, quaternary structure happens when we have 2 or more protein subunits combining to form that biologically active protein. And our hemoglobin example from the previous slide showed us we had 2 different types of subunits that would combine to form that overall protein by quaternary structure. Okay, so here's a visual depiction. So that primary structure is just how we're linking these amino acids together, and it's important, and as chemists that's probably our most attractive idea, but it's also a very simplified view of things because the primary structure doesn't tell us a whole lot about how the protein really looks or works. The secondary structure starts to form as a result of those amide linkages, the peptide bonds interacting with other peptide bonds along that chain, till finally now we get the tertiary structure, which is a result of the different side groups, the R groups there from our amino acid side chains interacting with each other. And then finally, we can have quaternary structure when we have those different tertiary structure subunits combining in a way that allows them to interact with each other in a functional overall protein. We used to be able to give phone cord examples for all this, but I'm afraid many of you out there are too young to remember the old corded phones that those of us old timers grew up with. So hopefully there's some other way that you can keep this straight. But if you think about it, if you do know the old phone cords, you could, pull them tight to sort of be the primary structure. Let them just form their normal coil would be more like the secondary structure. Then inevitably, if you ever talked on a corded phone, those coils would get coiled upon each other to form a tertiary structure. And ultimately, if you took a couple different phone cords, you could combine them into our quaternary structure. All right, so before we end our tertiary and quaternary level of protein structure lecture, let's have one final learning check. And this learning check asks that you identify the level of protein structure for each of the following. And then we have 5 different levels. Go ahead, read those levels, determine whether you think the level describes primary, secondary, tertiary, or quaternary structure. Stop the video while you make those assignments and start it back up when you're ready to check your work. Good luck. Okay, so let's sort of go in order here. So I'm looking at primary structure first. That's best described by B, where it's the order of amino acids in a protein. For secondary structure now, a beta-pleated sheet. A there is an example of a secondary structure type. Of course, we also had the alpha helix or the triple helix, but the beta-pleated sheet's the one that actually appeared here on this question. For tertiary structure, well, that could be the shape of a globular protein, as we described, or disulfide bonds between R groups. Once we're talking about the R groups, that's really at the tertiary level. And then finally, quaternary structure, we're going to want to see 2 or more peptide chains or subunits or some phrase like that indicating that this is a superstructure. It's made up of a couple different substructures to give an overall functional protein. So hopefully if you've been struggling with how you're going to differentiate these different levels, hopefully this learning check helped you to pull it all together. If that's not the case, if you've looked at the homework set questions and they're not much help either, please do get in touch with me. Let me know if there's anything I can do to help clarify. It is a difficult concept. I'm not going to sugarcoat that, but certainly there are some things we could do if you're having a lot of trouble and it's just not making sense. But hopefully that's not the case. Hopefully things are going very well and you're eager to get back to proteins and amino acids in the last lecture of this chapter. So can't wait to see you there.